Welcome to the new physiology course. As we will be discussing today, basic physiological concepts such as functions of the cell, homeostasis, and we'll talk about cell membrane, ion distribution, and body fluids. So, what is physiology? Physiology is a branch of biology that deals with the normal function of living organisms and their parts. And it's the science that studies cell functions with the whole human being at a chemical, physical, biochemical level. So, physiology is basically what the body or cell does, the function of the cell. Cells are the basic living unit of the body, and each cell is specialized to perform a particular function inside the organism. The body consists of cells that each has a function, like providing structure or taking in nutrients from food and converting them to energy, and each cell has its special function depending on its type and location in the body. An important concept in physiology is homeostasis. Homeostasis is the tendency of the body to maintain nearly constant range of conditions in the internal environment. So, homeostasis is basically keeping the environment stable. An example of homeostasis is the body temperature, which is maintained at around 37 degrees Celsius. When the body temperature rises above this level, mechanisms activate to lower the body temperature and vice versa. Blood glucose is also regulated. When glucose goes up in the blood, insulin is secreted to lower the body glucose, and when the glucose goes down in blood, glucagon is secreted to raise glucose level in the blood. The extracellular environment is everything outside the cell, and inside the cell we call it intracellular environment. These two environments are separated by the cell membrane. The cell membrane, besides its main function, which is separation, it has a role in cell recognition and in regulation in molecule movement in and out the cell. The cell membrane is mainly made of phospholipid bilayer and proteins, but they have different distribution of proteins on each side, which make it asymmetrical. For example, sodium level outside the cell is much more than the sodium level inside the cell, and the potassium level inside the cell is much higher than the potassium level outside the cell. Now let's talk about body fluids, the extracellular environment, which contains the intravascular compartment, which is the blood vessels, the arteries, and the capillaries, and also the interstitial compartment, which is between the cells, and the transcellular compartment, which surrounds specific compartments like the heart, the joint, brain, and abdominal cavity. The extracellular compartment contains 30% of the body fluids, and the rest is inside the cells. Fluid exchange between interstitial and intravascular compartments occur via capillaries. Now, to measure the fluids in each compartment, we use the formula concentration equals mass divided by volume. For example, if we want to measure the volume of water in a cup, we add a solute like sugar, and then we measure the concentration after it dissolves in the water. Finally, we calculate the mass of the water. We use the same concept for measuring body fluids. Just like sugar in water, the solute must be easily distributed. It should not be consumed by metabolism in the body, and it needs to go where we want to measure the fluid and not go somewhere else in the body. To measure the total water in the body, we inject radioactive water, which will mix with the body water, and by taking a sample from blood, we can measure the concentration of the labeled water the radioactive water, and then we calculate total body water. Now for the extracellular fluid, we use radioactive sodium ion because sodium ion does not enter into the cells. Now, measuring the intravascular fluid, which is the fluid inside the blood vessels and not between the cells or inside them. We use radioactive blood proteins like albumin because blood proteins remain in blood vessels and do not move out through capillaries into the interstitial compartment. Also, we can use labeled blood cells. Now, for measuring the interstitial fluid, which is the fluid between the cells, we calculate the extracellular fluid, then we subtract it by the intravascular fluid volume. Finally, we can measure the intracellular fluid by the formula total body water minus extracellular fluid. Also, we can use labeled potassium because potassium stays inside the cell and is not found outside the cells or in the blood vessels. Now, measuring body fluids is not always 100% accurate because water can be lost by urination and the solute must be fully dissolved and the solution must become homogeneous to give correct results.